Assalamualaikum warahmatullahi wabarakatuh. Today we are going to present about microorganisms in food production. So this is our group members in group 11. Aiman, Syakirin, Akmal, Nawarah, Aqilah, Nasreen, Farisa, Syazwina, Nabila and myself, Danial, Afiq. So we go through to the meaning of each term. A microorganism is an organism that can be seen only through a microscope, while food production is preparing food in which raw materials are converted into ready-made food products, either made from home or in industries. A little bit of history about microorganisms in food production. In 1837, scientists discovered the role of yeast in an alcoholic fermentation, meaning that it was microorganisms were involved in food production process. In 1860, a famous French chemist and biologist, Louis Pasteur, said that microorganisms are responsible in production of beer and wine. He demonstrated that heat destroyed unwanted microbes in wine and beer. After World War II, that the food industry began to develop the biotechnological techniques we rely on today to produce a wide variety of better, safer foods under controlled conditions. So there are 6 common types of microorganisms in food. First, bacteria. It is a prokaryotic microorganisms. It has a cocci shape, bacilli shape or vibrio shape. The second one is a mold, multicellular microorganism with mycelial, or we call it a filamentous morphology. It is normally withstand greater fluctuation in pH than bacteria and yeast. The third one is yeast. It is generally unicellular and differ from bacteria. It is grow at water activity of 0.9 to 0.94 and pH from 4.0 to 4.5. Yeast also can spread through air or other means and alight on the surface of foodstuffs. Next is the virus. It is cannot reproduce without a living host. It is too small to be visualized with an ordinary compound microscope, and it is also consists of DNA or RNA core surrounded by a protein coat. Next one is a protozoa. It is a parasitic reproduced by binary fission or multiple fission, cause many diseases in humans such as malaria. And the last one is a fungi. It is a eukaryotic organism that include yeast, molds, and mushrooms. It is found mainly in moist foods and need warm and moist places to grow. So these are 4 food products that use microorganism in production that we are going to present today. The first one is bread, the second one is yogurt, next is cheese, and the final one is beer, wine, and spirits.
Microorganisms in bread. Yeast, a specific strain of Saccharomyces cerevisiae, is used as a leavening agent in bread to produce gas, which causes the dough to rise, and it is added to the mixture of flour, water, salt, sugar, and shortening. Yeast cells create a small amount of alcohol and a large amount of carbon dioxide during fermentation. As the carbon dioxide bubbles are entrapped in the dough bulks up and become lighter and finer in texture. Saccharomyces cerevisiae, sometimes known as baker's yeast, is the yeast used in the production of bread. Other than act as leavening agent that contribute to the rise of dough for baking, yeast also contribute to flavoring and aroma. There are 4 steps in bread making, which are mixing, kneading, leavening, and baking. During kneading, air bubbles are built in the dough and they are assumed to be the early nuclei of the gas bubble, which will build up during the succeeding stages. During leavening, the metabolism of yeast chemically transforms assimilable carbohydrates into carbon dioxide and ethyl alcohol as the finished products. This slide shows different types of bread. Firstly, white bread. This bread is made by mixing moistened flour with yeast and letting it sit for several hours in warm environment. The yeast quickly ferments the sugar, producing alcohol, and carbon dioxide causes the bread to rise. Next, sourdough bread, where Candida malariae and Lactobacillus san franciscensis are the most essential bacteria in a sourdough culture. Yeast creates ethyl alcohol, which is released during baking, and carbon dioxide, which forms holes in the bread. Third is salt rising bread. Despite its name, salt is not a necessary component in this bread production because the bread is made with a rising agent that produced from a mixture of corn or wheat flour, milk, and potatoes that have been stored in very warm area overnight. The starter is said to contain Clostridium perfringens. This slide shows some chemical reaction involved in sourdough production. Proceed with back effects of microorganisms to bread. First is root spoilage, which occurs due to heat-resistant spores of Bacillus bacteria that have survived the baking process. Bacillus subtilis is the most frequent source. However, Bacillus licheniformis, Bacillus megatherium, and Bacillus cereus have also been linked to ropey bread. Fortunately, ropiness can be controlled by chemical or biological treatments. Example of chemical that can be used is acetic acid, while biological approaches include sourdough, liquid yeast, and lactic acid cultures containing Lactobacillus plantarum, Lactobacillus brevis, and Lactobacillus fermentum. Next, fungal spoilage occurs when visible mycelium appears on the product surface and it can emerge before the product shelf life expires. Fungi are responsible for changes in sensory properties of food stuff.
Next, we are going to talk about microbe in dairy product, which is yogurt. Basically, yogurt is made from a fermentation process, and there are three main steps that needs to be completed in order to create yogurt. The first step is to letting the bacteria yogurt to positively interact with each other, and the second step is to getting the right consistency, and the last step is to stopping the fermentation process by lowering the temperature. The first main step is to let the bacteria interact with each other. In a neutral, high-oxygen environment like milk, as thermophilus develop faster than L. bulgaricus. Thus, it takes the lead and it consumes oxygen and makes new chemical, creating the condition perfect for L. bulgaricus to begin its metabolism and thrive. L. bulgaricus is now in charge of breaking down part of the protein it make into amino acid. This makes it easier for S. thermophilus to obtain the nutrient it requires to continue to thrive. The second step is to getting into the right consistency. While bacteria strain absorb lactose naturally present in milk and convert it to lactic acid as they grow. The amount of lactose they convert to lactic acid determines how acidic the milk is. Casein or the milk protein begin to clump together as the milk becomes sufficiently acidic, changing the consistency of the milk to form a thicker substance which is known as yogurt. The final step is to stop the fermentation process by lowering the temperature. We must stop the yogurt fermentation process once the milk has converted into yogurt and we have achieved the ideal flavor and texture. Cooling it down will help as lower temperature will slow down the growth of the two bacteria that are deriving in the process. By preventing the yogurt from becoming more acidic, keeping it at lower temperature helps to preserve its textures and flavor. There are few people who cannot consume dairy products such as milk because they have lactose tolerant issue. Lactose intolerant is a condition in which people have symptoms due to decreased ability to digest lactose, which is a sugar found in dairy product. It is advisable for these people to consume yogurt as yogurt have the live bacteria that can help break down the lactose, so the body of those lactose intolerant people doesn't have to digest the lactose uh, by itself.
The next is the cheese production. Cheese is actually an accidental product obtained from milk. It is resulted from the contact of milk with rennet, the enzymes from ruminant stomach during storage. Therefore, the milk becomes separated into two, which are curd and whey. Next is the process of cheese making. The diagram shows the overview of processes during the cheese production. Jump on to the next is the involvement of microbes during cheese making. Microbes are added before the milk to enhance the coagulation process to the formation of cheese. They are also used for salting and a finish or storing of the cheese. During the separation process, lactic acid bacteria and bacterial enzymes such as rennet are added to milk. Milk becomes sour due to bacteria and coagulate the casein in milk. Therefore, it resulted into separation into curd and whey, which cheese is made up from the curd of milk. The ripening process of cheese is actually divided into two, which are the decomposition of curd and fermentation. During the decomposition of curd, microbes synthesize lactose into lactic acid, while proteolytic enzymes from Brevibacterium linus help to break down the protein. As the penicillin rock forte produces lipase, it releases short-chain fatty acid, which also helps to release flavor of cheese. Whereas during the fermentation, bacteria from Propionibacterium ripen lactic acid and produce propionic acid, carbon dioxide, and also acetic acid. The acids produced give flavors into the cheese, while the carbon dioxide form an eye-like host, such as at the Swiss cheese. Last from the cheese is types of cheese. There are actually many types of cheese used in the world. Examples of the frequently used is cheddar, gorgonzola, ricotta, cottage cheese, mozzarella, and also cream cheese.
Microbes in food production: beer, wine, and spirits. Beer and wine are made by fermenting sugary juices. Spirits, such as whiskey, gin, and rum, are made by fermenting juices and distilling the fermented product. Beer brewing process: to make beer, cereal grains, usually barley, are malted to increase the concentration of starch-digesting enzymes that provide the sugar for fermentation. Malted grain is crushed and mixed with hot water about 65 degrees Celsius, producing mash. After a few hours, a liquid extract called wurst is separated from the mix. Hops, which is flower cones from hop plant, are added to the wurst for flavoring, and the mixture is boiled to stop enzyme action and precipitate proteins. A strain of Saccharomyces is added. After fermentation, the yeast is removed and the beer is filtered, pasteurized, and bottled. Fermentation process: fermentation produces ethyl alcohol, carbon dioxide, and other substances, including amyl and isoamyl alcohols, and acetic and butyric acids, which add to the flavor of the beer. Most wine is made from juice extracted from grapes, and the juice is then treated with sulfur dioxide to kill any wild yeast that may already be present. Sugar and a strain of Saccharomyces are then added and fermentation proceeds. When fermentation is completed, liquid wine is siphoned to separate it from yeast sediment and, if necessary, cleared with agents such as charcoal to remove suspended particles. Spirits: spirits are made from the fermentation of a variety of fruits, including malted barley to make Scotch whisky, rye for rye whiskey and gin, corn to make bourbon, wine or fruit juice to make brandy, potatoes to make vodka, and molasses to make rum. Distillation process: distillation separates alcohol and other volatile substances from solid and non-volatile substances. Strains of Saccharomyces are the fermenters for all alcoholic beverages. Many different strains have been developed, each having distinctive characteristics. Both the organisms and how they are used are carefully guarded brewers' secrets. Which alcoholic beverages have higher alcohol content? Because of distillation, the alcohol content of spirits ranges from 40 to 50%, much higher than the typical 12% for wine and 6% for beer. Wines do not contain more alcohol because when the alcohol concentration reaches 12 to 15%, it poisons the yeast carrying out the fermentation.
So the last part is conclusion. What we have learned, microorganism is really quite related in food production. There are thousands of different types of microorganisms everywhere, in air, soil, and water, in the digestive tract of animals and humans, which has its own pros and cons. Microorganism is important to food production, not just food production, important to food safety, processing, preservation, and storage. Microbes such as bacteria, molds, and yeast are common use in the manufacturing of food and food ingredients such as cheese, beer, bread, and dairy products, just like already stated before. So here let's look how important microorganisms rather than the microbial contamination in food. First, microorganisms in food production helps the manufacturers to increase the food quality. For example, yeast in bread production make the dough expand and rise then become soft. Just like the gardenia bread, as the taste really good and soft because of yeast. Next, microorganism also helps in food texture, make the various type of product really good to consume. For example, lactic acid bacteria make the milk can be a yogurt. In rough, the bacteria make the milk become acidic as coagulate with the protein, then come out with yogurt. Other than that, microorganisms also important as rapid growth, contained growth, so minimum space is used, predictable product is made under controlled conditions, and they may grow on waste materials from other processes. Microorganisms such as bacteria, molds, and yeast are the most common microorganisms that cause food spoilage. And microorganisms also that we need to know widely used in the production of food and food products. Food manufacturers continue to use microorganisms today to make a wide range of food products. For example, fermentation. Fermentation not only gives food a good taste, texture and smell, but it causes changes that reduce the growth of unwanted food microbes. This improves the food's storage life and safety. Last but not least, new discoveries and technological advancements are always being made, implying that the use of microorganisms in food production and the benefit that come with it are not restricted to what has been shown thus far. That's all from us, and thank you.